Hey, good morning, everybody. It's Monday, March 23rd, and you are watching the screencast video for AP Bio. This is chapter, I don't even know what chapter this is, 48. We're going to the nervous system. Hope you guys had a good weekend. And before we start, just a couple of important things. Last night I sent out a survey to everybody. You guys may have heard about some changes to the AP test, and I'm still in the process of figuring everything out. I don't really want to make too many comments about it because there's still some stuff I don't know for sure, and I know a lot of you guys have questions, and so I've emailed admin and the district. I'm still waiting to hear back from them. But for sure, once I get that information, I will relay that to you guys. I hope to have something for you guys by the time we get together tomorrow for the live stream. Yeah, I also keep checking your email because I think for tomorrow's live stream, I may try to use Zoom. I just think it'd be cool for everyone to see each other. For the results that I have gotten back so far with the survey, a lot of people are feeling really isolated, and I don't know how much you guys are actually getting FaceTime and stuff like that, but it really does help to just see other people. They don't have to be your best friends or um, people you're super close to. It's just nice to kind of recreate that social environment. My wife and we've been doing like FaceTime dates or meetings with like family members and random friends and people we haven't talked to in a while, are texting and wanting to check in, and we do probably 3 or 4 or even 5 of those a day. So it's been a really good way to supplement the social aspect that's definitely missing right now. So I hope you guys are doing okay. We're at a full week into this shelter in place now, and um, it's kind of a good time to check in and see how the last week has gone for you. Some people are seemingly doing okay, dealing with the changes, and they've kind of made a routine and a structure for themselves. And some people have not really had that structure, and maybe by now they're really kind of starting to feel it. The survey results, I got some of them already. About half of you guys have responded, and it sounds like a lot of you guys miss school, which is just nice to hear. And I'm with you guys, man. Being at home is, I thought it'd be kind of nice. You get to, I mean, we're definitely sleeping a little bit more, which is good, but it's not the same. Definitely miss you guys. So, all that to say, tomorrow maybe we'll try out Zoom. Just check your email. I'll try to set something up for us so that we can see and hear from everybody. Because the live streams are cool, but I like to see. I get to hear from you, but I like to see you guys too and see how you're doing. Um, so yeah, even with all the changes and stuff, as we're still figuring it out, I've kind of been reflecting over the weekend. I totally understand that a lot of you guys are feeling kind of down and disheartened about all of this, and even though there hasn't been any like official communication from the district about like what's going to happen after spring break, if you're reading the news at all and you're seeing what's going on in other countries, this shelter in place thing is probably going to be in place for a while. So that also means we're not going to be able to go back to school as soon as we had hoped, or if at all. And so that's raised a lot of questions for people. That's it's kind of a weird feeling to have that, I don't know how to say this, that finish line, I guess, that you're all chasing, kind of taken out from under you. And so we're gonna have to dig deep and kind of reflect and be honest with ourselves about why we do what we do. I know for all of your lives, as long as you can remember, you've been motivated by pretty much external things, right? You have to be in school, you have to get good grades, you have to go to college and all that. And maybe one of the good things about all of this is it makes us think about why we do what we do, myself included. When I'll be honest, I do keep track of what's going on YouTube, and I logged in, don't know, Saturday morning, just to see how many people had watched the last screencast, and it was definitely a lot lower than earlier in the week. And I totally understand, especially seniors. I know you're going through a lot right now. This is such a crappy way to finish your high school career, and I hope we can get creative in like looking for ways to celebrate you guys. And again, if you haven't been FaceTiming people, even if they're not your best friends, like just connect with people. But anyways, yeah, I know it's been hard. So seeing the YouTube video viewership drop, and maybe YouTube gives me too much too much access to the analytics. I can see what the average time that people have watched it is, and for a 40 or 50-minute video, the average view time being like 10 minutes, it's kind of depressing. So it's made me look at my own motivations for doing things, and it's like, I guess it's kind of like a tree in the forest thing. Like if a tree falls in the forest, then you won't hear it. So the new question is, if an AP Bio teacher makes a screencast and no one watches it, is he still making videos? Is he still teaching? And it's kind of a deep existential question for me. So I'm still figuring that out. But in the time being, it's encouraging to see that those of you guys that have responded in the survey is saying that these things help. And at the end of the day, that's what I want for you guys, is to be a resource to help you understand this stuff. So I'm going to keep going. I'm going to keep doing my job. And, as you guys sort it out, if you find your way coming, back here and start watching again, that's great. But if you don't, that's okay too. I guess we're just kind of figuring this out together. So I'm going to try to keep this one shorter so that you have more reason to keep watching. But we're going to go into chapter 48, which is the nervous system. And we're actually going to be doing that for most of this week, not just this chapter, but the next one as well. And it's a personal favorite of mine because this is what I studied in college. But I hope you guys enjoy it and get something out of it. Again, fill out that survey if you haven't already. And I'll take a look at it, taking any information that may be coming from the district or admin, and I'll be sharing that with you guys tomorrow. So try to set an alarm and be up at 9:30. You can tune in from bed if you need to, but I'd like to have a little family chat about what's going on with the class. All right, let's get started. So chapter 48 is the nervous system, and what we want to do in this these next couple lectures is understand how the cells use their methods of communication, right? The stuff we covered on Friday to create this amazing network of communication for your body, which is the nervous system. So over here. So the just maybe some review for some of you guys, but the functional unit of the nervous system, the cell that makes up the nervous system, is called the neuron. And you have millions and millions of neurons in your body. From an evolutionary standpoint, you recall that we started off, with cnidarians of having that nerve net, and then as we moved onto land and we became cephalized and bilateral symmetry became a thing, these neurons started to migrate towards different parts of the body, especially towards the head. And with cephalization, you have the creation of the central nervous system. And so these neurons all link up together. They're not physically touching, but they do create this network that makes up your nervous system. So let's take a look at some of those parts. Like all cells, the neuron also has a cell body. This is basically where you would find all the major contents of the cell, the nucleus, ER, mitochondria, all those normal things. Now from that, you have these extensions of the cell membrane that make two kinds of fibers. One set are these little tiny fibers that are really highly branched. These are called dendrites. And I want you to think of dendrites as kind of like the ear of the neuron. This is where neurons receive messages. This is how they hear from others. And then those messages kind of make their way from the dendrites into the cell body. And then when the cell body decides, hey, this message is important enough, it's going to send it off to the next neuron through this one long fiber called the axon. And you can see the axon both in length and in structure has some different features to it. We'll talk about what some of those are in a bit. Actually, one of those features is these little white blobs around it. This is called the myelin sheath. Myelin is actually made out of each of these little bulbs. Morning, hun. Actually made out of a cell, and depending on what part of the nervous system we're looking at, there are different kinds of cells. But for now, you should know that the myelin sheath is basically a fatty substance that wraps itself around the axon, and it acts as an insulator for the electric message that's firing down. So you can kind of think of it as like the plastic coating on your charger cables or whatever. It just keeps the electricity insulated and keeps that signal strong. At the very end of the axon, you have this area called the axon terminal, and that's where the message is sent out. Terminal meaning like the end of something, right? So the message gets sent out of these little axon terminals at the end. One thing I should mention before we move on is you'll notice that in this picture there's actually two neurons. So this is one neuron right here, and then this is another neuron here. And so how do they connect? The axons of one neuron are going to meet up with the dendrites or even the cell body of the next neuron. And just some key terms that we use for this. So the connection here is called a synapse, and you'll see that later on in this slideshow. But the neuron that comes before that synapse, the one that's sending the message, is called the presynaptic neuron, and then the one that receives the message, that's the postsynaptic neuron. So here's kind of an overview of all the different parts that we talked about. The cell body, axon terminal, dendrites, and the myelin sheath. And this picture here just kind of shows you that there's a lot of different ways that neurons can be arranged, depending on if it's a sensory neuron, an interneuron, or a motor neuron. They do vary in their structure and function. Okay, so in addition to neurons, you have another type of cell that doesn't really get a lot of credit, but it's super important. These are called glial cells, and glial cells are the supporting cells for your nervous system. They provide the structure, they provide the myelin sheath, actually, in some cases. So let's take a look at some of these. The first type of supporting cell is called an astrocyte, and it gets its name because they kind of branch out in every direction like a star. That's where we get the word astro from. And these astrocytes provide structural support from neurons, and kind of create a framework or a scaffolding for all your neurons to sit on, so that your brain isn't just like this goopy mess. It is really, really soft. If you ever get a chance to dissect a fresh brain, it's incredibly soft. It's like Jello. But if it weren't for these astrocytes, it would just be almost like this fluid-like substance, really soft jelly. So astrocytes provide that support, and they also help regulate concentrations of ions and neurotransmitters. And we're going to see today that ions are incredibly important to allowing for these neurons to work. The other type of cell are the oligodendrocytes and the Schwann cells, and they basically are the same kind of cell, but for some reason they give them two different names depending on if it's the central nervous system or the peripheral nervous system. I'll talk about those in a little bit. But what they both do is they make this fatty myelin sheath, and the way that it works is these cells are kind of like big flat slabs, if you kind of imagine a piece of dough that's been rolled out, and then what it does is it kind of wraps itself around like a jelly roll or a cinnamon roll around the axon down here, and it creates these bundles. So you can see in the cross section that kind of spiraled shape, that would be that myelin sheath that's been wrapped around. Okay, and then this photograph here actually shows a real picture of what that looks like. So again, what they do is they insulate the signal and they keep the signal strong. There are these little gaps between these myelin sheaths called nodes of Ranvier. They also speed up the movement of the signal. Actually allows the signal to jump from one node to the next to the next. Okay, so I mentioned CNS and PNS. What exactly does that mean? That's referring to how the nervous system is organized. And the way it's set up is you start with nerves, right? So these are bundles of neuron fibers, and these are kind of the most basic structures of our nervous system. And again, we saw these back in our evolution unit with even early invertebrates like cnidarians and platyhelminthes and the worms. They were growing these nerve fibers which bundled together hundreds, if not thousands, of neurons and allowed them to fire messages off to different parts of the body. And that way, these organisms can not only sense what's around them, but they can also regulate their body in response to that. We also, if you remember, we had ganglia. Ganglia are clusters rather than like running a long, thin fibers traveling over long distances. Ganglia are these clusters of neurons that are capable of carrying out pretty simple calculations. And even in early invertebrates like the flatworms, Platyhelminthes, the ganglia kind of served as their brain. You know, they didn't really have high brain function. They couldn't necessarily think, but they had some basic pre-programmed functions like maybe recognizing food or mating behaviors that were kind of pre-built in, and the ganglia would manage these. Now, as we evolved to be more complex, these ganglia are going to start to cluster together, especially in the head region. And so that's really what the brain is, right? If you think about the brain, it's just like a bunch of different clusters of neurons, and that's why there's different regions of the brain. We'll talk about that later this week. So ganglia are kind of a precursor to that. Now, we also have ganglia in our spine, and I'll give you one example. There's a pretty cool ganglia in the lower back called the central pattern generator. It's abbreviated CPGs, and they will regulate things like walking. So, you know, when you walk across the room, you don't, your brain initiates the thought like, I'm going to walk, and you should take that first step. But as soon as you start taking that first step, the other leg kind of follows it. I mean, you even watch babies when they kick, they automatically click left, right, left, right, left, right. And the reason for that is because you have these ganglia, these clusters in your spine that regulate walking. And it doesn't actually involve you using your brain. I have not been able to find this picture, but back in college, I learned about this experiment. It's pretty cruel, but I think you guys will like it because it's cruel. They got either a cat or a dog, and they were studying these central pattern generators, which are in the lower back. And so what they did is surgically, they cut the spinal cord of these animals just slightly above the central pattern generator, and that caused this cat to basically be paralyzed from the waist down. So the back legs didn't work. And so now you have this cat like dragging itself around on the front two legs, right? It's really sad. But the cool thing was they put this animal on a treadmill, which sounds so mean, but the scientists grabbed the back legs with their own hands and they turned the treadmill on, and the scientists actually like moved their back legs, manually, and after a couple of steps they let go, and to everyone's surprise the back legs kicked on and started walking. Now, it's not because the brain is telling the legs to do that. It's because the central pattern generators in the lower back, even though it's cut off from the brain, it was receiving messages from the leg muscles that the muscles are working, and the central pattern generator was like, oh, it's time to walk, I'm going to just turn that on, and the legs were moving on their own. So I know I'm getting a little off topic, but it's a pretty cool example of how ganglia can control really basic functions. So here's kind of what we talked about, like kind of the evolution of our nervous system here. And really just to get to the main point here, we have the nervous system is broken up into two parts. You have your central nervous system, which you need to know are made up by your brain and your spinal cord. Okay, this is like where all the main instructions are sent out from. This is also where all your sensory information goes into to be processed, so that you know what's important, what's not important. Now, extending from the spinal cord and also from the brain, you see all these smaller nerves that are branching off. Those nerves are carrying information to and from your central nervous system to the rest of your body, right? And the word we use in neuroscience is we use the word innervate. So those nerves are going to innervate your muscles, they're going to innervate your heart, they're going to innervate your digestive system. And they're also going to bring information from the heart and the digestive system and your skin to your central nervous system for processing. So all these nerves that are outside of the central nervous system, they're referred to as your peripheral nervous system. We abbreviate that as the PNS. And that's just going to be the nerves stemming off the spinal cord. But there are also some, you can see if you look closely here, there are some nerves that stem off the brain directly. Those are technically part of the peripheral nervous system, and those are called the cranial nerves. And those are really important nerves that innervate your eyes, your nose, your mouth. One of them, called the vagus nerve, is actually exits the brain and goes straight down to the heart, and it plugs into your pacemaker. Remember that SA node that we talked about. So how do we process information? This is a very simplified, but it's a big picture big picture of how information is sent around your body, right? And what we have here is a picture of what's called the reflex arc. We like using the reflex arc as an example because it's just super simple. You guys know that when we have a reflex, you do something without thinking about it, right? And the reason for that is because a lot of our reflexes are actually managed by neurons in our spinal cord rather than in our brain, and that's why you can respond so quickly. But that also simplifies things enough for us just to understand a simple example of a neuron or neural network. So the way we process information is it always starts with sensory input, right? Whether you're seeing something, tasting something, hearing something, or feeling something on your skin, it always involves some kind of stimulus around you being detected by your body. And that sensory input is managed by a sensory neuron. These sensory neurons we'll look at in the next chapter are always hooked up to some kind of receptor. So on your tongue, it's a chemical receptor. In your eye, it's a photoreceptor. In your skin or your muscle, it's a stretch receptor. And when these receptors are triggered, that causes the sensory neuron that they're partnered with to fire off a what's called an action potential. That's also called like a nerve impulse. That if you look closely, that causes the message to move, in this case from the leg, up the nerves and into the spinal cord, which this is a cross section of the spinal cord. So you can see the sensory neurons. In this case, we're looking at like a hammer hitting the knee, but this could also be used stepping on a nail or something. But the sensory neuron takes that information and brings it to the central nervous system, right? So this all right here, that in the outside, that's the peripheral nervous system, and here's that central nervous system. Now, once we get to the central, we have to process that information, and that's done by, I gotta zoom back out to show you the next thing, is by the integrating neurons, and that's done by an interneuron. In this picture, the interneuron is represented by this grain neuron. And I know it looks really short, so I guess technically it is shorter than a sensory neuron because it's staying in the spinal cord, but it is really important because it's going to process that information and then decide, is this important enough to respond to? And if it is, the interneuron is going to communicate with the neuron that's going to send the message back out to tell the body how to respond. And that's referred to as motor output. This is executing a command from the integrating center, right, from the interneuron saying, okay, body, here's how I want you to respond. And so when the doctor hits your knee with that little rubber hammer, how does your body respond? Your leg kicks, right? But the kick, you actually have to have a message from the central nervous system telling it to kick. And that's done through this red neuron right here, which is called the motor neuron. So motor neurons get their name because they innervate muscles, and they tell muscles when to contract. So it doesn't matter if you're getting hit on the knee, or you're playing baseball, or you're playing piano, or even right now while I'm talking, I'm moving my tongue, I'm moving the muscles in my mouth to create speech, and those are all motor neurons that are being controlled by the central nervous system. So that's what we call that a reflex arc. But what I really want you to get out of this is that this is the pathway on how we process information. Okay, and so just kind of like maybe be aware of that. Think about as you go through your day, what are all the times where you see or hear or feel or taste something, and then you respond to that. And anytime you do that, you see kind of this three-step process of sensory input, integration, and motor output being involved. Now, of course, there are many other pathways that can form, but this is just one simple example that's kind of easy to wrap our mind around. Now, the last thing I want to talk about today are how neurons actually generate these impulses. Okay, because it sounds simple just to like have them fire off a message, and I will warn you, like this gets a little bit detailed. It's so detailed. I took a 10-week class just on what we're about to talk about here in the next couple of slides. So I'm going to try to, for the interest of time and just keeping it simple, I'm gonna try to simplify it as much as I can. But keep in mind, this is actually a really complicated process that even after 10 weeks, I was just barely starting to understand it. Okay, so if you get kind of bogged down by the details, just keep coming back to the big picture. The big picture is neurons can fire off nerve impulses. Okay, and the way we fire off those nerve impulses, which is kind of measured in the form of electricity, is actually by moving ions around. So let me kind of break it down for you. In college, we actually played with this piece of equipment a lot. It's a voltage recorder, and what it does is it allows you to measure the voltage difference between two sides of the membrane. Okay, so we would actually get an axon or a neuron from a what's it called? A giant squid. And the reason we do that is because the squid axons are some of the biggest in the world. Like they're actually like, half a centimeter wide or something like that. So you would take two electrodes, two little pins, and put one on the outside of it in the outer liquid, and you would poke one into the inside of the axon. And then this machine here would measure the voltage difference between them. Now, why is there a voltage difference? Because in a living neuron cell, there is a difference in ionic gradients across the plasma membrane. Okay, I know that sounds really fancy, but here's basically how it's set up. And I have I'm going to switch over to this little animation here. So here's the deal. Okay, make sure I get this right. So in a neuron, we have two ions that we want to keep track of. Okay, sodium and potassium, and all the sodium is found in the outside of the cell. Okay, so using sodium pumps and things like that, or active transport, the sodium levels are really, really high outside, and potassium levels are really high inside. Okay, now from what you know about concentration gradients, what does that mean? That means all the sodium outside wants to rush into the inside of the cell. Okay, whereas all the potassium inside wants to rush outside of the cell. Now, I know they're both positively charged, and I'm not going to get into the details of this, but for purposes of our discussion, just understand that the concentration of solutes, including sodium on the outside, cause the outer portion of the membrane to actually carry a positive charge. Okay, and you can see that in this picture here. So the combination of sodium and other ions outside, and the combination of potassium and other ions and other solutes inside cause the outside to be positively charged and the inside to be negatively charged. And so when you measure that difference, that's kind of what we were doing with that device, is we have an electrode on the outside and an electrode on the inside. And when you measure that difference, you get a reading of some sort. And that reading always comes out in the form of an electrical signal. So in this case, in this picture, you can see here that they're measuring negative 70 millivolts. Okay, and that's what we call the membrane potential. So depending on where those ions are and what their concentrations are, that potential can change. Now the reason that's so important is because that again means that the neurons are more negative inside compared to the outside, and you can see there's some other ions here that can contribute to that membrane potential. Now what that means is that in this moment when a neuron is not firing a message, there is a membrane potential of negative 70. Okay. Now, what creates an electric current is when that membrane potential changes. Okay. So I'll give you a little spoiler alert. When we go from negative 70 to positive 65, that represents a electrical charge. Okay, a electrical impulse that's formed. And so how do we get it to go from negative 70 to positive 65? Well, keep in mind, what's keeping it at negative 70 right now is that we have all this sodium on the outside and all this potassium on the inside. So how do we get them to switch? The charge to switch, we just have those chemicals switch. Okay, we're going to have all the sodium move to the inside and all the potassium move to the outside. Now you guys know that sodium and potassium are both ions, which means they can't go through cell membranes. And that means we need, to help them move, we need ion channels to open and close in response to stimuli. Okay, and these ion channels, we talked about this in the previous chapter, are gated ion channels. So as long as those gates are closed, sodium and potassium cannot move, and the membrane potential stays the same. But as soon as they open in response to some kind of stimuli, they will switch places. And that's kind of what this animation shows here. Let's see, I'm going to try to, there it is. Okay, so here what this shows is when I push play here, I'm going to turn the volume off on that. Here are the gated channels, right? The purple ones are the sodium channels. So when they open, sodium is allowed to diffuse from the outside of the cell into the inside of the cell. And this is happening super slowly in this animation. In real life, this would happen like in milliseconds, okay? And then shortly after that, the potassium channel, they're kind of walking through all this, potassium channel is going to open, and the same thing is going to happen here, is that the potassium is going to rush from the inside of the cell towards the outside of the cell, okay? And when it does that, that causes the charge to switch, okay? Now, we'll put that all together in a bit to kind of show you what that means. Now, I want to mention really quick before I move on, what causes these ion channels to open are really three kinds of stimuli. Number one is a stretch-gated ion channel. These are really found in your skin and your muscles when your cells are physically moving or stretching. All right, because like you're moving your muscles or someone grabs your arm, that can cause ion channels in your skin to open and create a nerve impulse from your hand to your brain so that you know that someone's grabbed your arm. Another example is a ligand-gated ion channel. That is, we talked about ligands in chapter 11. That's when a chemical binds to the ion channel and that causes it to open. And then the most common one in our neurons are a voltage-gated ion channel. So that's where the gate only opens if there's an electrical change nearby. Okay, and that's the one that I really want to focus on right here because it's kind of like a chain reaction, right? What I mean by that is when the sodium and potassium over here move, the outside right now is positive, the inside's negative. When they move, those charges are going to switch. The inside's going to be more positive, and the outside's going to become more negative. Okay, and that represents a change in voltage. Let's see if we can kind of see that happening with this simulation here. So here you have your neuron. This is what your neuron looks like kind of at its resting state, okay, when you're not using it. It's positively charged on the outside, negatively charged on the inside, and what allows for that is that all the sodium has been pumped outside the cell and all the potassium is pumped inside the cell. As we move forward, watch what happens here. The sodium channels open, okay, and when they open, all the sodium rushes in, and that causes a reversal in the charge in this section right here. Okay. Now keep in mind, this picture doesn't show this, but there are potassium and sodium channels along this entire membrane, on the top part of the axon and on the bottom part of this axon. And you can see what's happening in this video is that the purple part, which represents the electrical impulse, is kind of moving to the right. Okay. The reason why it's moving, I'll kind of let it play here, towards the right is because all along here, these channels, these are voltage-gated ion channels. Okay, so these channels right now in the yellow section, those gates are closed. What's gonna cause those gates to open is if there's a voltage change nearby. Okay, so because these channels right here opened and they created a change in the voltage, that's gonna cause the channels next door, right next to it, the ones with the gates here, they're going to open. Okay, right here and right here. And when they open, then sodium and potassium switch places, and the charge is going to flip from positive outside, negative inside, to positive inside and negative outside. Okay, so you can see that when one section of the neuron goes through that switch, where the ions are allowed to move, that creates a voltage that then triggers the next section of the neuron to open its gates, and then that causes that part to switch, and so on, right? So the result of this is that you end up having, I really don't love this animation. The one I really like, I actually left it on my teacher drive at school, and I cannot find it at anywhere online. I looked all morning this morning and last night for it. So this is not the best one. But what you can see here is as this chain reaction happens, it's kind of like people doing the wave at a baseball game. You know, when one group does a wave, the next section, they do the wave. And the result of this is that you get this movement of this electrical impulse from one side of the neuron towards the other. Okay, and that is called an action potential. Okay, that's also referred to as a nerve impulse. So when I say that you're firing off a message from your brain down to your hand, that's what that actually is. That's an action potential, and that's caused by the movement of sodium and potassium in and out of your cells. So I know that's not the best, maybe not the best explanation of it. There are a lot of great videos on YouTube that kind of like animate it and show you how it happens. So if you get a chance, maybe watch that. If I think of it, maybe I'll post some videos here. So here's kind of a diagram to show like what that looks like. Oops, sorry. So when we're measuring these voltages, you know, with the device that I was showing you earlier, what we see whenever an axon fires a message is this big spike. Okay, now this spike is similar to, but not the same as like a spike you would see on a heart monitor. It's kind of different, but you would see this spike. And what's happening during the spike is this: at -70, the neuron's at rest. Okay, so let's say you're not thinking about something right now. I have a word in my head. I'll say it in a bit. You're not thinking about this word right now, which means that neuron that's responsible for firing that word is not thinking about it. As soon as I say it, though, you're going to fire a message, okay, from one neuron to another neuron. And that word is purple elephant. Okay, I'm assuming none of you guys were thinking about purple elephants right now. So as soon as I said that, the part of your brain that regulates the thought or the words purple elephant fire off a message. And so what's happening there is something called depolarization. Okay, we're going to go from a resting state. Oh my gosh, sorry, I'm struggling. There it is. Okay. A resting state where you can see that most of the sodium is on the outside, most of the potassium is on the inside. But as soon as I say purple elephant, the sodium channels only, not the potassium channels, but the sodium channels are going to open, okay, and those sodiums are going to rush into the cell, and that's going to make the cell switch from being positive outside to and negative inside to negative outside and positive inside. Okay, and that's called depolarization, and this is probably made by some British people, so they use an S there. We would use a Z, depolarization. Okay, so the voltage-gated sodium channels open. That's immediately followed by the opening of the potassium channel, which causes the potassium to rush back out, and that kind of restores our membrane potential. So we go back to being slightly positive outside and negatively inside. And you can see that in the graph here. So here's that spike, that's the sodium rushing in. Here's the spike going back down, that's the potassium going back out. And then you can see where we actually overshoot. Okay, we went past -70, and then we're going to come back to our resting threshold right here, or resting state, which is when the neuron's not thinking about purple elephants. And how do we get all those chemicals back to their sides? This is where we use a sodium-potassium pump. So this is actually a form of active transport using an ATP. All the sodiums get pumped back out, and all the potassiums get pumped back in. And this cycle here, these 4 steps, these are the 4 steps of an action potential. That's one spike, okay? And when we're measuring the impulses of someone's brain, the spikes actually end up looking more like, I'll come back to this, like this. These are actual readings of action potentials. And you can see every time you see one of those lines, that means an action potential has been fired down that axon. Now, depending on what message can be sent, you can see different patterns, right? These are almost kind of like barcodes. So if your brain sends this, where it goes, and it fires all these really closely sequenced action potentials, that is sending one kind of message, whereas this one here is a little more spread out, and this one's more sporadic, and all those are different messages. And why is this so cool? Because nowadays with technology and with our capabilities in measuring brain waves, you can see this scientist here at the University of Minnesota is actually controlling this robot, moving these cups around, and he's actually able to do that. Oh gosh, sorry. Struggling here. Keep pushing the wrong button. There we go. So he's actually moving these cups with his brain just by thinking it, because the electrodes pick up on those action potentials, the computer deciphers it, translates it to code for the robotic arm here, and then the robot arm is actually able to grab the cups and move them onto different levels. And this has huge implications for people, especially if they have paralysis. We now have people, of course you have to have the money for it right now, but people can turn on the lights in their house, they can check their email, they can do really basic things by just thinking it, with having these electrodes on their head measuring these action potentials. Okay, so I don't want to spend too much more time on this, but and you can read up on your own. If you have questions, you can ask about during the live stream tomorrow. But why is this so cool? Because this is the language of your brain. Okay, by simply moving around sodium and potassium in and out of your cells, and then creating these different patterns, these are literally what your thoughts are. Everything about your brain and your person, your personality, your memories, your emotions, your ability to learn, your ability to communicate and move your body and all of that takes place with the language of these action potentials. And it's all made possible because we're doing active and passive transport of ions into your cell, which is crazy if you think about that. So that's kind of your little preview to an action potential. I can answer more questions if you have any, but I know there's a lot you're probably wondering, but this video is already 40 minutes long, and if I make it any longer, no one's gonna watch it, so I gotta move on. The last thing I want to go over really quickly is the synapse. Okay, so as I mentioned earlier, neurons don't actually touch each other, and so there's this switch of language that has to happen, right? What we just went over right now is the language in the that's spoken in the form of electrical impulses, right? These action potentials. But when they arrive at a synapse, because the neurons aren't actually touching, there's a gap there. That's called the synapse. The message actually gets changed to a different language, and that language is a chemical language. And we talked about this in chapter 11 with neuron and synaptic signaling, right? So I can go over this kind of quickly. Action potentials cannot cross a synapse, and so when the depolarization of the axon arrives, the action potential arrives at the very end of the axon terminal, what it does is it causes voltage-gated ion channels to open, but this time it's not for sodium or potassium, it's for calcium. And so the elevated calcium levels triggers the release of neurotransmitters. And here's kind of how it happens. When the electrical impulse arrives here, and the picture doesn't really show that, but imagine the electrical impulse coming down and it arrives here, the channels here sense that change in the voltage, they open their gates, and calcium rushes into the inside of the axon terminal. Waiting nearby are these vesicles filled with neurotransmitters. Right? These are chemical messengers, and these neurotransmitters, when they are exposed to calcium, will migrate and move to the end of the axon terminal. They will fuse with the membrane and do exocytosis, and those neurotransmitters spill into this gap here called the synaptic cleft. There's that word right there. And those chemicals then bind to receptors on the other side. Okay, so this is an example of short distance signaling. Okay, kind of like paracrine signaling. Now, this big blob on the other side here, this is actually the dendrite of another neuron. Okay, so this neuron is receiving the message that there's a bear coming at you, this bear is chasing you. So they need to be able to fire off a new electrical signal. And so how do they do that? Well, in the membrane here, you have the receptors, but if you look closely, the receptors also look like ion channels, and that's because they are. Okay, these ion channels here are not voltage gated, so they don't open when the voltage changes. These are ligand gated ion channels, so they only open when a chemical binds to their little receptor site and it opens. And so this is what this picture shows here, is that when the neurotransmitter binds to this this ligand gated ion channel, the channel opens up, and that allows for sodium and potassium to move, and that generates a new action potential. It generates a new voltage change, and you don't see it in this picture, but nearby there would be other voltage-gated ion channels that would then respond like that wave that we talked about at a baseball game. They would open, and then that would start a brand new cycle of action potentials. Okay, so this happens every single time one neuron is communicating with another. Okay, and so we have this like alternation of like an electrical message, chemical message, electrical message, chemical message, and they kind of alternate, and that's how you're able to send messages throughout your entire nervous system. Okay, so I'm going to wrap it up here. We're going to stop here. The next, the reading guide, if you choose to do it, and also the next slide kind of talks about the different kinds of neurotransmitters. You've probably heard of some of these before. Acetylcholine is the one that is used oftentimes to communicate with your muscles, to tell your muscles to contract. You've also heard of dopamine and serotonin. These are really important neurotransmitters for our brain's reward systems to tell you like, hey, this is good for your survival and reproduction. You should keep doing it. And there's a bunch of other ones down here. We're not going to worry about them too much. Okay, so I hope that's a good preview. If you need more information, you can dig around on YouTube. There's a lot of other videos that have really great animations, and you can also just make a note of it and ask me during our office hour things tomorrow. And again, I'm going to try to set up something on Zoom so that we can see each other for that. It's probably a little bit better than the live stream. Okay, hope you guys are doing good out there. It's 10:27, so this should be uploaded in the next like 30 minutes, and hopefully you guys will be awake by then. Check it out, and hope you have a great day. Take care, guys. Bye.